today I would like to talk about the QC tool that was recently integrated in our PixOnline 11. So our webinar agenda can be divided into 6 parts. The first, I would like to talk about the QC attributes in proteomics, and I would like to take you a tour about our PixOnline QC feature. And following for that, I want to use 3 examples to show you how we can use our tools in such a large cohort study. And finally, I'd like to demonstrate our PixOnline QC software and to give you a thorough understanding about what we are doing in here. Okay, let's talk about the proteomics first. We all know proteomics is a very complex process and it involves in multiple steps. For example, we start from sample preparation, we digest our sample with enzyme that we like to choose, and we got our peptide. The peptide will be separated by RC, and those separated RC was injected into mass spectrometry and detected by mass spec, and finally we get our data so we can analyze and generate a great story for the science. But we all know in all this process, every step can go wrong and bring you the sample, bring you the variety in the final result, and we have to find a way to validate our data. So for doing this, there are multiple attributes. We can use them to do quality control in our study. And I just want to spend some time to quickly go over this. So the first one that we want to check probably is your sample preparation. In sample preparation, especially for bottom-up proteomics, we would like to do digestion. We can choose any enzyme that we need. One thing we need to watch is my enzyme specific, which means that is my enzyme cutting at the right amino acids. So we need to check this specificity ratio. What's the percentage of my peptide is cut at the right amino acid? Following is my enzyme efficient? There are multiple cutting sites on a protein. So is my enzyme cut in every cutting point? We also need to watch that. So missing cleavage ratio. Did I enzyme miscut any a lot in my experiment? Once we have our peptide, we are going to elute them on RC. So we need to also take a look at our RC setup. So for RC elution, so when we talk about RC, peak is our main word here. We can see there are many peaks shaped, and we really only want a nice sharp peak, not too wide, not too sharp. So only by doing this, we have a nice distribution of our data collecting point. So for checking this, we need to check the full width of our peak and the half width and the base peak chromatogram. So to ensure, okay, our RC is behaving properly, our peptide are separate based on our design. Once the peptide got separated by RC, they're getting into the mass spec, and we also need to check the mass spec QC attributes. So for mass spec detection, we collect a lot of data. Like in here, we're showing you a 3D shape. So in this very rich data, we want to answer the question is, does the mass spec detect enough features? Is my data, or in short word, is my data good? So there are a few things that we would like to watch, which is the MS1 numbers, MS2 numbers, features divided by precursor ratio, which means how many detect features were generated from all the precursors that we use, be peptide ID numbers, protein ID numbers. All these are important to verify is our data a really good data. So up here, we talk about almost roughly 10 attributes that we need to watch. For building such a QC steps can be very time consuming in our daily research. So for solving this issue, for solving this, PixOnline provide an integrated QC tool that I'm going to show you here. It can help you get a much easier process to validate your data. So here is a overview of the QC. Our QC was fully integrated in our regular data search flow. You can see we provide a DDA, ID, DDA quantification, DIA, ID, DIA quantification, and all these things are fully integrated. You don't have to do anything extra besides a few clicks. So here is a setup window. As you can see, you can simply check this box, and then you can start select your control sample. You can set up your own accepted tolerance, plus minus 10. We'll set up here. Also, if you have any standard peptide that you would like to monitor in your QC process, you can also put in here. So, so here is the setup, and I would like to take you through the QC feature in Pix Online, so you can have a good and you can have an idea about how we choose to organize data and how we're going to analyze data. For the QC attributes that we watch here, basically cover all the topics that we talked about earlier in our first 5 minutes. So we cover the sample preparation, we monitor the misclipping ratio, enzyme specificity. For the LC, we monitor the full width of the peak, half width, the base peak chromatogram. For the mass spec, of course, we monitor the mass spec arrow, and as well as all the other identify numbers like the protein peptide identifying numbers, quantifying numbers. All this are can be chosen to monitor based on the user's needs. So let's go through how we're going to present data. Here is a general overview of the attributes status that you select to watch. For example, in here, you can see your control samples and what your setup for the accepted tolerance and how many samples you choose to watch here. And if the sample you choose and its attributes all fall in the range that you set up previously, we consider it's a pass. And we provide a ratio of the pass sample. Of course, if the attributes that did not pass, in another word, fall out of the tolerance range that you set up early, we consider that's a fail. And how many samples have at least one failed attributes was listed, and what kind of the total number of the failed attributes is also listed. And on the side, we provide a bar chart to give you an idea about, okay, what your failed attributes are in all your samples. So in here, we give you a overview about how your sample behave, quick answer about did your sample pass or not. So I hear you probably want to also see some detail about, okay, what my sample is actually doing. So for doing this, we provide a systematic analysis of your data. So we can definitely detect the variance in your samples. So for example, for all the attributes, we give out a table that lists, okay, what part of your attributes are failed, and their real numbers, any numbers that was fall out of the range, or in other words, fall out of the tolerance level will be marked as red. And to give you a direct view of your attributes matrix, you can simply click on any column, and you will get a line chart to give you a more direct view of your attributes, matrix of the attributes. We also provide a TNC chromatogram to help you to have a quick view of your samples run. You can select any sample to be presented here. Not only we can do variation, we can also check the sample reproducibility. We provide the Venn diagram to see what kind of overlap between the samples, the Pearson correlation chart, your quantification relationship between any two samples. Plus, we also give out the retention time shifting box plot here to tell you how your LC behave. The mass spec arrow violin chart, as if you are doing quantification, a CV chart is showing also to help you to get okay, what's the variation between your quantification. So we talk about this data. They're all more general. There's no really targeted. We can also do targeted analysis in the QC feature of the Pix Online. So we can take a look at that. So for targeted, which is also the standard peptides that you choose to watch, we give out a comparison chart, reproducibility chart here. So you can see the retention time shifting of your standard peptide, the match-chart ratio shifting in your standard peptides. So up here, I hope you already have a general understanding about how our QC feature is organized and what kind of data we're delivering. And we want to spend the rest of this webinar to get some case study so you can know, okay, how we can use this to help you go through this QC process. For large sample cohort, that's recently getting more and more widely used in the clinical study. For example, this is one project was done in 2021 with some COVID-19. There are over 128 patients, and there's another project was done in 2019. There's over 1,500 samples that was collected to monitor the obesity process in patients. All this was done due to for any clinical study, there's the large sample variance, and we have to use a large sample size to compensate this sample variation. And since the sample size got large, and many times the research time was dragged into long period, we have a desperate required to perform an easy and quick QC process. So in here, I want to use some data from this project to give you, let's mimic a QC process of a large sample cohort study. So in this work, over 1,500 plasma samples are separated into 17 plates. And on the plates, the samples are injected randomly. In here, we pick 20 of them to let's see, okay, what kind of QC we can do in our QC feature in Pix Online. The first thing that we are doing is to set up a control sample that was can be set in the setup page. Also, you can pick up any samples that you would like to watch. In here, all 20 samples are selected. But let's imagine if you have a much more sample list, you can use a built-in search to get a quick selection of the data you want to watch. And we will show this function again in the demonstration. On the QC attributes, you can select any attribute that you would like to monitor. In general saying, in proteomics, the people seems more interested with the identified protein groups and peptides. So in here, we also select this to let's monitor some of our samples. Let's see how they're behaving. So we already see this sort of figure already. So here is the result of this set of data. You can see, okay, this is my control sample. My tolerance is set up as 10%. And my total sample count are 20, 16 of them pass. So, okay, pretty good ratio. It's over 80% passed. I still have 4 failed samples. And what are they failing? And they seem to all have a low peptide number. All right, so now I have an idea about, okay, what kind of sample, what's the percentage of my pass sample, and what's the percentage of my failed sample, and what attribute they failed. They seem to all focus on these peptide numbers. But how good is pass? Yeah, we say we passed, but are they only barely meet the threshold? So we may want to take a look. So here is a comparative view of our control sample and the average of pass samples. So in here we can see in our control samples, there are over 6,300 peptides, but on the average of our pass samples, we are nearly reached to 6,500. So we are saying, all right, so my pass sample is beyond at least a threshold. So my pass sample is good. But what I'm doing in my failed sample? So in here, we can see, okay, in all my samples, we have a low peptide numbers. And if there's an RNA field, it will be also listed in red. But in here, it's just all peptides, low numbers. So we up to here, we get the idea about what kind of the attributes are fitting in my 20 samples. But what's the issue? What caused the low peptide numbers? We have to try to figure out where this demand is coming from. Then we start to get into data to see, okay, what is the issue? Where is the issue coming from? So here is a table of our LC-MS raw data attributes. You can see not only we have, so in here we list out all the LC attributes that was monitored in LC in our PixOnline QC module. So you can see we have a precursor not only in 4 samples, actually they extended to I think it's 8 samples. So that means the we have some issues in our sample runs, and but where they coming from, we are seeing okay. So in our full width, this attributes starting from plate 4, we have a increase. So that indicate, well, it's very possible our LC system that have a problem at least. We can plot this one into the line chart and to give you a more direct view about your data, since sometimes data is hard, may give you some misleading. So in here, you can see the.
Did I just put in an IT ticket for access to IDBS and then automatic?
So in here, we can see there is a precursor line chart here. You can see, okay, from the plate 4 injection up to 6, we have a low precursor one. Actually, this trend starting from the plate 3. So we may, so this problem is only below threshold in this run, but may also extend to further upstream. And in here, you can see in plate 4 run, we have a clear jump of the full weight of our peaks. Therefore, all right, so now you have a very clear view. Okay, so we have issues on the LC. At least we are going to look at that direction. We all know even do experiment, things may not get lucky, and occasionally many errors can show up at the same time. Can something others go wrong? In here, we give out a TIC chart from we are selecting our control sample as well as to this failed samples. We can see their TIC level. We can from this we can see from their TIC chromatogram, there's no major difference. At least we probably see something missing in the first 5 to 20 minutes, but no more than that. So at least we can tell, okay, at least sample get injected. It's not our sample injector issue. Could it be? Is there anything else? We may want to check our mass spec as well. We are seeing some low attributes in plate 4, but we all know this already. It's coming from a identified low precursor that leads to a series of low numbers, for example, PSM, peptides. We all know this already. That's okay, but its mass spec goes wrong. Could mass spec cause this issue? We can see, okay, in our mass spec attributes, all our numbers are fairly consistent. Therefore, it's unlikely. Furthermore, we give out a violin chart of our control samples, pass samples, and fail samples, their PSM and error distribution. We can see between the pass sample and fail samples, their mass spec error are very similar, same as the fragment error. So now we can tell, okay, MS is not causing a problem in my run. Now we are going through some data in the untargeted analysis. Can targeted analysis provide more evidence? Here is the evidence from the standard peptides. This is the retention time reproducibility of the standard peptides. On the bottom, that's our samples. And in here, in all these numbers, they are listing how many times a standard peptide is not observed in your certain samples. And if you click this I button, you can change the blank into this small bar. It tells you, okay, where your sample cannot observe certain standard peptides. From this chart, we can see, okay, we have a clear shift of the retention time of all my standard peptides starting from plate 4 to up to plate 5. Now we can say, okay, combine what we know from the previous slides, we can clearly say this problem is focusing on the LC part. If I'm doing the experiment, I'm starting checking if there is any leaking, any column pressure increasing, so I can identify my problem and solve that. So we talk about, up here, we talk about how to use our Pixel Online QC tool to monitor the real sample in the large cohort clinical study. But we all know we also run QC samples in this kind of study. So I'm going to show you next is how to evaluate some QC samples. In this work, you organize an international collaboration study, and across the whole world, 11 international laboratories was chosen to run run the same samples to evaluate whether BIA can be implemented into the clinical study. In this work, QC sample was run in 3 back-to-back way before running the real sample, which was sample A and sample B. And in here, even though the actual study was run in the DIA, but in this 3 run, they are all DDA. So we are using DDA QC sample to evaluate our machine status. And I would like to use some data from this project to give you, to demonstrate how we can evaluate our QC runs and evaluate my machine status. So you see this sort of form before. This is we are taking out data from lab fab from day 1 to day 9 and all their QC samples. You can see in lab fab in day 7, replicate 3, we are seeing a series low attributes. Okay, but I'm only seeing this once. If I'm doing this thing, I may wonder, okay, is this a one-time thing or actually something happens? Now, then we can plot some attributes into a line chart. We can take a direct view. Sorry, so in DDA, we are plotting this one into some bar charts. Now we can see I have this peptide number low on all my lab, all my day 7 runs. Even besides of one of my run is below the threshold, there's 2 other runs are just barely meet the threshold. So looks like there's something wrong in my day 7 runs. From our previous case, let's take a wild guess. Could it also be LC issues? Now we are plotting our half-weight metrics in the bar chart. We are seeing in all my QC samples, my half-weight of my peaks is looking great and seems to be behaving well. In here, we have a cumulative precursor count and versus retention time. We are seeing all my runs, all my QC samples are overlapping almost perfectly, and that means my precursor are separate accordingly. It's all well behaved. Also put the TSC chromatogram between day 1 and day 7. We list them here and just give a comparative view. But up to here, we are not seeing anything bizarre. All right, that didn't answer our question. So could it be something else? And we can take a look at other data. So I like the LC issue. Our mass spec. So this is PSM arrows that the violin chart was plotted, and all the samples are presented here. We can see, okay, even we have some drifting. We have some mass spec accuracy drifting, but unlikely to be an error source. So we are seeing all this arrow are still fall in the plus minus 5 ppm range, which is normal. Error tolerance was set up in our software analysis. So all right, not even mass spec. But from the fragment arrow, we are seeing some interesting thing. We can see in here, not in this runs, not only we have a high error in our MS2, we are also seeing, okay, the distribution is uneven. So that suggests, okay, our error is not on the detector. It's actually probably somewhere else. Maybe the fragmentation chamber. So in this paper, actually at the end of the day 7, the HCD chamber, the error routing multiple was maintained, and you can see on day 9 the issue was fixed. So now we can see the PIX Online QC can also help us to using the QC tool to evaluate our machine status. For doing large cohort study, the reproducibility is also one major thing. We want to check if our data can be successfully reproduced on our current setup. Our QC tool can also help with that. So here is some data taken from this paper. In this run, 1 microgram of HeLa sample are injected into a Explorer 480 3 times. So the system reproducibility is tested. And from here, we can compare our attributes. We are seeing all the attributes are in the range and they are distributed evenly. We can plot them into a bar chart to have a more direct view. You can see everything very consistent for doing a label-frequent quantification. The CV plot of this set of data is excellent. It's only 4.7 at 4.17. Now we know at this moment, all this my machine is in prime state. We are ready to do some serious experiment to collect data. For checking sample reproducibility, like we mentioned early in our webinar, we provide also a identification Venn diagram for the quantification. We provide a Pearson correlation chart for peptides and protein. For any this set of kind of comparison, you can choose any 2 samples that would like to compare and put them in here. You can see at this moment, my peptide protein quantification correlation are excellent between my sample 1 and sample 2. So if you are doing ID, we also provide the data completeness chart. So in here, you can see in this set of data, the data completeness is over 88%, which means that over 88% of the protein can be identified in all of my 3 samples. So which prove the machine is excellent, is in great status. I just want to highlight this part. So our QC module also provide a capability to monitor the sample preparation process. So in here, you can see we also monitor the mixed cleavage ratio, the enzyme specificity ratio. In here, you can see, okay, am I choosing the right enzyme, or is my digestion condition is well suited for my project? You can, from this 2-1, you can see, okay, did I perform a successful experiment, not only by for experiment design, but also to control your sample preparation process in your large sample cohort study. Just for your convenience, all of our chart can be exported into PDF version, and our chart on the web page can be zoom in and zoom out to suit the user's need. And once you zoom out your figure, you can also export the zoom out version of your figure to save in PDF to for your better organizing in project. And we can export our data into Excel versions, and so you can, by a simple click, you can export your data into extra file, so you can either collect them or put them into any tools that you would like to organize your data record. Okay, that's the end of my my talk. So up here after 30 minutes talk. I hope you have a general idea about how we are using, how we design our QC tools. It was fully integrated in the database search, quantification, and everything is in one workflow. And all the static analysis are summarized into one report for you to use, and any data figure can be exported, and the whole process is automatic, systematic, it's customizable to your needs. And I'm going to do a software demo and to show you how our data is actually put in use. So one moment. So here is a setup of our label-free quantification. In the previous part, you set up your data searching parameters, you upload data. I'm not going to do too much about this since there will be another webinar in next week. I hope you can all join to take a look at our DIA workflow. So at the end of all these setup parameters, you can simple by checking this QC box to activate this function. So you really don't have to do anything extra to start this process. And for the control sample, you can select any sample you would like to use as a benchmark, or if you don't have a perfect one, you just want to have an idea about how some behave, you can even select your average sample. For the accept tolerance, it's changeable. You can type in the accept tolerance, or if you like, you can use the arrow to change up and down of your accept tolerance. And if there is any standard peptides that you would like to use, you can put in here. It's a very simple process. You have your peptide sequence, you have a chart state, and just copy and pasting here, and then you're good to go. Once everything is done, you click submit. I'm not going to do it here since the analyze is done at this point. I'm just going to give you the direct report, how they look like. Here is the summary view of our attributes. You can see this is something what you see before in our talk, but I want to go over this filter tab first. So in here, you are seeing what kind of the attribute that you would like to watch, what kind of sample that you would like to be involved in this QC process. If just in case you have a very large sample size, you can use this search button. For example, in here, I would like to only to check the sample from plate, let's say plate 3. So just simple click plate P3. It's right there. You can just select them all or deselect them to make things easier for you. Okay. And for the attributes, you can select them all or by click this, or you can only select a few ones that you would like to monitor, and that's also doable. And click save, and this page will automatically update for you. And you don't have to do the whole analysis again. It's very fast and convenient. We already talked about this, so I won't go into too much detail about that. Just to let you know, okay, we can export our file by single click, and you will have a PDF version pop out in your downstream window. Sorry, I lost my mouse. Oh, here it is. You can, by single click, you can export your PDF, and in here, you are seeing your past label frequent attributes, and you can see your failed one and how their attribute distributed. And it may not be only one, so every one is highlighted with a red dot. And also, you can by simple click in here, you can export the static information about your failed and past samples. This is a organized the table and the Q's and the figures tab. So we divide this one into two parts. The first one is more about the LCMS. As you can see, you see this kind of chart before. You are seeing MS1, MS2 numbers, and any column can be further plotted into this kind of line chart to give you a better direct view. You can see we can plot the full wave, you can plot your half wave. They can all be automatically generated there. You can also export this figure into a PDF format for your so easy. Maybe you need to populate this data, so that's easy for you to generate any figure. You can also zoom in, zoom out on this one. As simple as I'm using Windows. I'm still using scrolls to give a zoom in, and maybe also to give a zoom out on the X and Y axis. And once you think, okay, I had enough, and you can click 1-to-1 and bring everything back to the initial state. Here we give our TIC chromatogram, and you can select a sample. Same idea about your about what we showed in the earlier in the search button function. You can also search in here. Let's say you only want to watch P1 played, so you can have P1, and also we have P12 here. But you can select anyone. Let's say we want to keep our P1, but I want to update them to also compare some from the P2. So by simple click, okay, here you have that. This part is the end of our LC-MS part, and in here we are focusing more about the library search or database search. You can see we started giving out besides something you may see before, the MS2 numbers, precursor numbers, we are seeing some PSM numbers, identified peptides, quantified peptides, MS arrows, your ID rate. Again, they can all be plotted into this line chart here. And at the end, you can see your reproducibility of your sample. For example, in here, a Venn diagram, pick any one you want to monitor. It will update automatically for you. And as the Pearson correlation chart, you can also pick any two that you want to check. Very convenient to use. So in here, we give out a box plot of medium data RT. You can check your peptides if there's any major retention time shifting compared to your control sample. And in here is our MS1, MS2, MS arrow. Your samples are separated into different groups, pass and failed. Also for quantification purpose, we give out a CV value of your protein. So on this page, it's all about untargeted analysis. And for standard peptides tab, we give out the targeted analysis. The first one you see this is a retention reproducibility chart. For this number is how many times the peptide missing. You can back click that. You have to see, oh, I missed my run in most of my missed my peptide in most of my runs. And I can zoom in to see, okay, which one am I actually missing? For example, this one has also missed the EMP4 of some of my samples. So you can have your direct view about, okay, how my standard peptides behave. For the mass-to-target ratio, here is one here for your mass-to-target ratio reproducibility of the standard peptides. You can see how my mass spec behave. Similar logic, we have the unseen numbers with plotting in here, and you still can zoom in to be more focused on one peptide. So we are seeing, okay, okay, there's some fluctuation about my peptides, but in general, it's still quite good. It's only, let's say, plus minus 0.003, something like that. Okay, that's the end of my webinar and my demonstration, and I'm happy to take any questions.